Welcome to Roadmap to Residency Evidence-Based Applications. I'm Abhiraz, and in this video, we will be discussing about amino acid metabolism. Let's first talk about amino acid breakdown. So there is no storage form of amino acids. So unused amino acids needs to be get broken down. So when amino group is removed, we have ammonia plus alpha keto acid. We have to remove this ammonia. So ammonia is toxic to body and it is transferred to liver in a non-toxic structure and converted by liver to urea, which is also non-toxic for excretion. So amino acid breakdown. So usually the first step is removal of nitrogen by transamination. The amino group is passed to glutamate. So the alpha ketoglutarate is converted to glutamate. So amino transferases. So they transfer the nitrogen from amino acids to glutamate, and they all require vitamin B6 as a cofactor. And the two used as liver function test that we commonly use perform as a lab is an alanine aminotransferase and aspartate aminotransferase. So these are all these form of enzymes which are used to transfer nitrogen from amino acids towards glutamate. Okay, so this is aspartate aminotransferase, which we have used to transfer the amino acids or to transfer the nitrogen from amino acids towards the glutamate particle. So there are two methods for transfer of nitrogen from glutamate to liver for excretion in the urea cycle. It can be through glutamine synthesis or through alanine cycle. So talking about glutamine, so glutamine is a non-toxic molecule and it transfers nitrogen to liver for excretion. The glutamine synthetase is found in the most tissues. The glutamate is converted to glutamine by glutamine synthetase, and this glutamine is going to transfer the nitrogen to liver for excretion. Okay, so this glutamine molecule just transports to liver, and it is used as a mode of transportation of nitrogen towards the liver. In liver, the glutamine is converted back to glutamate. So that's how you know that glutamine is only used to transfer nitrogen. Um, so you can see that the glutamate is converted via the glutamate dehydrogenase in the ammonia and alpha ketoglutarate. So the alanine cycles, we have talked that the method of transfer is either through glutamine or through the alanine cycle. So alanine cycle is used by muscles to transfer nitrogen to the liver, and the glutamate nitrogen is converted to alanine by the enzyme alanine transferase, right? So the alanine to liver, it gets converted into pyruvate, and the nitrogen is transferred back to glutamate. So we can see that uh, we have transfer through alanine transfers the nitrogen from the glutamate towards the alanine, okay? And again, the nitrogen is transferred back to glutamate. So the nitrogen is removed from glutamate by the enzyme glutamate dehydrogenase again in the liver after that. So we can see that in the muscle and the liver, there exists a cycle. Now this is only thing that you need to understand the alanine cycle. If you have even forgotten everything that I have told you about the alanine cycle, you can see that the ammonia gets attached to the glutamate. So the reaction is that there is transfer of ammonium from glutamate towards alanine. This alanine get transferred towards the liver, and again this alanine gets converted to glutamate, right? And again this glutamate by the enzyme glutamate dehydrogenase, there is excretion of there is excretion of ammonium, and again the glutamate is left out. So the urea cycle, so the ammonia, the ammonium ion is converted to urea, and then this urea is excreted into the urine. The urea is synthesized from ammonia, carbon dioxide, and aspartate. So you give ammonia, you give carbon dioxide, and you give aspartate, and then there is formation of a urea molecule. The first reaction, the first and second reaction of the urea cycle is in the mitochondria. And the first reaction itself is a rate limiting step, which is what? Is that the ammonium gets acted upon by the carbamoyl phosphate synthase 1. So ammonium gets attached with carbon dioxide to form carbamoyl phosphate. And so N-acetyl glutamate is an allosteric activator, carbamyl phosphate synthase 1, and the enzyme will not function without this cofactor. So the enzyme carbamyl phosphate synthase 1 will not function without this enzyme N-acetyl glutamate. Okay. Now it is synthesized from glutamate and acetyl-CoA. So in case of increased fed state or in case of increased protein state, there is increase in the N-acetyl glutamate because there is increased production of ammonia. So it is used to regulate the urea cycle also. And there is the pyrimidine synthesis. So the glutamine gets attached with CO2 by the enzyme carbamyl phosphate synthase 2 to form carbamyl phosphate. Again, the second reaction also occurs in the mitochondria. Now we have formed carbamyl phosphate earlier in the first reaction. So the carbamyl phosphate gets attached with ornithine by the enzyme ornithine transcarbamylase to form citrulline, right? Now, the ammonium gets attached with CO2 by the enzyme carbamyl phosphate synthetase 1 to form carbamyl phosphate. This carbamyl phosphate gets attached with ornithine to form citrulline. Now, this citrulline again can transfer from mitochondria towards the cytosol. Okay, so here we have the citrulline in the cytosol. Now, in cytosol, what happens that the citrulline combines with aspartate to form arginosuccinate, and this arginosuccinate gets converted into arginine with the excretion of fumarate, and this arginine gets converted to ornithine again back. So ornithine can move out and in of the mitochondrial membrane. So the ornithine gets converted back to its own nature, and there is production of this urea. Now this urea gets excreted into the urine. Thank you for watching. Keep studying.